So if you lived north of the 50th parallel, you used to be safe.
Right. Historically speaking, at
Yeah,
least.
exactly. That was this sort of invisible line on the map. But nature, unfortunately, doesn't really care about our maps.
No, it really doesn't.
Thanks to migratory birds acting as like literal international flights for infected ticks, we're now dealing with an unstoppable virus. And this thing has a mortality rate of up to 40%. It's just bypassing border controls entirely. So today, we're taking a deep dive into the bizarre ecological nightmare that is Crimean-Congo hemorrhagic fever.
It sounds like the premise for a disaster movie, honestly, but it is a very real, very present issue.
Yeah.
We're looking at a pathogen that causes anywhere from 10,000 to 15,000 human cases globally every single year. And that geographical footprint you mentioned, it's actively expanding as we speak.
Which is exactly why we've pulled together an incredible stack of research for you today. We've got a really comprehensive new clinical review from the 2024 journal Infezioni in Medicina, along with the absolute latest fact sheets from both the WHO and the CDC.
It's a lot of data, but it paints a very clear picture.
It does. Okay, let's unpack this.
Yeah.
Because, to understand how you or I could actually catch this, we have to look at the delivery mechanism. I mean, this isn't a flu. You don't just breathe it in on the subway, right?
No, far from it. The primary villain driving this entire disease network is a specific type of hard tick. It's called the hyaloma tick.
The hyaloma tick.
Right. And historically, the virus was geographically boxed in by this tick's natural habitat. So we saw it primarily in Africa, the Middle East, Asia, and parts of southern and eastern Europe.
Below that 50th parallel.
Exactly. That 50th parallel north we mentioned, that was essentially the ecological temperature limit for the tick to survive. It just couldn't handle the cold up there.
Wait, so if the tick can't survive the cold, how on earth is this thing spreading so fast now into new territories?
Well, it's all about the birds.
The birds.
Yeah. The virus has essentially hacked into global migration routes. The birds themselves don't actually get sick from the virus.
Oh, really?
Right. They just act as long-haul transport. So an infected tick latches onto a migratory bird in, say, North Africa. The bird flies thousands of miles across continents, and eventually the tick just drops off into an entirely new territory.
Wow. So these birds are just like airdropping infected ticks all over Europe.
Basically, yes. For example, Spain has seen around 15 cases since 2013, which is a direct result of this tick expansion.
That is wild. So if you're hiking in Spain right now, you might be walking right into a newly established tick population. But hold on, if the birds are dropping these ticks into new fields, what happens to the animals the ticks bite? Like, do cows and sheep get this hemorrhagic fever? Do we just see deer dropping dead in the woods?
That is actually the most insidious part of this entire cycle.
Yeah.
No, they aren't dropping dead. When these infected ticks land and bite domestic animals, cattle, sheep, goats, you name it, those animals absolutely get infected. The virus enters their bloodstream and it circulates there for about a week.
But they don't get sick.
Not at all. They act as silent reservoirs. They don't show a single obvious clinical symptom.
They just go about their day like nothing happened. That's terrifying.
Completely asymptomatic. And this creates a devastating tick-animal-tick cycle that goes entirely unnoticed by farmers or, public health officials.
Because you can't see that the cow is sick.
Exactly. An uninfected tick bites that infected cow, drinks the viral blood, becomes infected itself, and the cycle just perpetuates silently in the grass.
Right. Just waiting until a human steps into that
Yeah.
grass. But before we talk about what happens to humans, there was this one detail in the WHO data about an abattoir, like a slaughterhouse in South Africa, that just blew my mind. The ostriches.
Ah, yes. The ostrich anomaly. It is a really fascinating outlier.
It's so weird. You've got cows and sheep walking around totally fine, and most birds are completely resistant, like you said. But for some reason, ostriches are highly susceptible to getting sick.
They are the exception to the rule, yeah.
Right. And there was this major outbreak documented at an ostrich slaughterhouse because the workers were just being exposed to the infected blood of the birds.
Which is incredibly dangerous.
Yeah, it actually forced a massive regulation change. So if you're processing ostriches now, they have to be kept in a tick-free quarantine for 14 days before slaughter, just to make sure the virus has cleared their bloodstream.
Which perfectly illustrates the two distinct ways you can catch this disease.
Okay.
You are either bitten directly by an infected hyaloma tick, or you have direct contact with the blood or tissues of an infected animal. Usually that happens during farming, slaughtering, or butchering.
Okay, let's follow that path. Let's say you're an agricultural worker, or I don't know, you're just on a hike and you get an unlucky tick bite. The data outlines a timeline for what happens next, but the incubation period seemed totally inconsistent to me.
Well, it depends entirely on your entry point.
How so?
If your exposure was through infected animal blood, say, you had a cut on your hand while butchering, it typically takes about 5 to 6 days before symptoms appear.
Okay.
But if you are bitten by a tick, the incubation is incredibly fast, usually just 1 to 3 days.
Here's where it gets really interesting, though. Why would a tiny tick bite be so much faster at making you sick than getting raw infected blood directly into an open wound? I mean, that seems backwards.
I get why you'd think that, but it comes down to the biological delivery system.
The delivery system.
Right. A tick bite isn't just a passive needle puncture. The tick actively injects saliva into your skin, and that saliva is packed with compounds designed to inhibit your local immune response. And this is key. Stop your blood from clotting.
Oh, wow. So it's like a burglar who doesn't just pick the lock, but also cuts the phone lines to the alarm system.
That's a great way to put it.
It's actively laying out a red carpet for the virus.
Yes, exactly. It prepares the site for the virus to enter and replicate immediately without your immune system fighting back right away. Direct blood exposure, while highly dangerous, obviously, doesn't have that chemical assist from the tick's saliva.
So the virus takes a bit longer to establish a foothold on its own. Okay, and once it gets that foothold, you start feeling, what exactly? Because the early symptoms described in the research sound frustratingly generic to me.
They are very generic. Sudden fever, severe headache, muscle aches, red eyes.
Right. If you walked into a clinic with that, any doctor would just tell you that you have the flu.
They absolutely would, but there is one very specific, visible early warning sign. They're called petechiae.
Petechiae, what are those?
They are tiny red spots that often appear on the roof of the mouth or on the skin.
And that happens because the virus is going after a very specific part of your body, right? The endothelial cells.
Yes.
I'm trying to picture what those actually are.
Right.
Are they like the walls of your veins?
Close. Think of the endothelial cells as the innermost lining of your blood vessels.
Okay, so if the blood vessel is a pipe, the endothelial cells are like the plumbing tape sealing the inside.
Yes. And the virus acts like a saboteur that sneaks into your system and specifically degrades that plumbing tape. And that degradation leads directly to the hemorrhagic phase, which usually hits around day 3 to 5 of the illness.
So really fast.
Very fast. As the vascular permeability increases, meaning, as your pipes start to leak, your immune system realizes what's happening and goes into an absolute overwhelming panic.
It freaks out.
Exactly. It triggers a massive release of pro-inflammatory cytokines.
Ah, cytokine storm.
Yeah.
I think a lot of people listening probably remember hearing that term constantly over the last few years. It's when your immune system basically goes scorched earth to kill a virus, but ends up destroying your own body in the process.
Precisely. In the case of this specific virus, that cytokine storm triggers a horrific chain reaction. It leads to something called disseminated intravascular coagulation, or DIC.
Okay, English, please. What is DIC actually doing to you?
Well, normally, if you have a leak in a blood vessel, your body sends platelets to form a clot and plug the hole, right?
Right, just normal scabbing.
But because the virus has degraded the lining of your vessels everywhere all at once, your body panics and starts forming millions of microclots all over your circulatory system.
Oh, God. So it's trying to plug a million microscopic leaks all at the same time?
Exactly. And in doing so, it uses up almost all of your platelets.
Wow.
So now you have no platelets left for normal clotting. The result is severe, uncontrolled bleeding. Patients suffer from massive bruising, bleeding from the gums, nosebleeds, and uncontrolled bleeding at any IV or injection sites in the hospital.
That sounds like a nightmare. The review mentioned that if they test your blood at this stage, the viral load is basically the ultimate predictor of whether you survive.
It is the defining metric, yes.
Like if you have over 10 to the 8th or 10 to the 9th copies of the virus per milliliter of blood, the odds of a fatal outcome skyrocket. Your organs just shut down.
Right, the sheer volume of the virus overwhelms the cardiovascular system. It also heavily targets the liver and spleen, causing them to fail alongside the massive blood loss.
But, and this is wild to me, 60 to 90% of people do survive this, which is incredible given what you just described.
It is a testament to intensive care, yes.
But we have to talk about what happens after. Because surviving isn't just, walking out of the hospital a week later and getting lunch. The recovery, the convalescence takes up to a year.
It's a very long road.
We're talking hair loss, extreme fatigue, polyneuritis, which is severe nerve pain. And the stat that really stopped me in my tracks was from a study in Turkey. almost 48% of survivors exhibited symptoms of post-traumatic stress disorder.
When you think about the clinical picture, it's tragically logical.
Yeah, I guess so.
You go from feeling perfectly fine to having flea symptoms to suddenly bleeding from your gums and nose while rushed into intensive care, just watching your body fail in real time. It is profoundly terrifying, violent disease to endure.
So if you're a doctor on the front lines and a patient comes into your ER in that hemorrhagic phase, their internal plumbing is leaking, they have no platelets, and a cytokine storm is raging, how do you actually fight this?
The most sobering realization in current medicine is that there is no magic bullet.
Really? None?
None. The absolute foundation of treating this is purely supportive care. You are essentially trying to keep the patient's body running manually while their own immune system tries to clear the virus.
That sounds incredibly stressful for the medical staff. You're just constantly managing their fluid balance, monitoring their electrolytes, and pumping them full of donor platelets and fresh frozen plasma to replace everything the DIC is destroying.
You're trying to outpace the bleeding, yes.
But surely we have antivirals. Like, both the CDC and the WHO bring up a drug called ribavirin. But then the primary clinical review we dug into explicitly calls the use of ribavirin controversial.
It does.
So what does this all mean? Why would public health agencies recommend a drug that scientists consider controversial for a disease with a 40% mortality rate?
It's a classic example of how messy medical data can get during outbreaks. Ribavirin is a broad-spectrum antiviral. Its goal is to target the RNA replication of the virus.
Okay, lost me again. What does that mean for the virus itself?
Think of it as jamming the virus's biological photocopier. It tries to stop the virus from replicating its genetic material.
Okay.
And early observational studies looked incredibly promising. For example, a protocol used during outbreaks in Iran showed that oral ribavirin had an 80% efficacy rate in confirmed patients.
That's huge.
Right. The data suggested that if you gave it within the first 48 hours of symptoms, it dramatically reduced mortality.
That sounds like a massive success. So where is the controversy?
Well, the controversy emerged when researchers finally had the time to do rigorous, large-scale analyses. A major meta-analysis back in 2011 and further comprehensive reviews later on revealed that the actual quality of evidence supporting those early ribavirin claims was surprisingly low.
Oh, no.
Yeah. And then a non-randomized controlled trial in 2022 looked at a very specific metric. Did ribavirin actually reduce the viral load or the viral mutation rate compared to patients who only received supportive care?
Let me guess. It didn't.
The rate of viral clearance was essentially identical in both groups. The drug didn't seem to be doing the heavy lifting everyone thought it was.
That is incredibly frustrating. You think you have a weapon, and it turns out it might just be like a placebo effect wrapped in early panic data.
Exactly. It's tough.
But the research did highlight a combination therapy that actually worked, didn't it?
Yeah.
Something to do with steroids.
Yes. And this represents a massive shift in how we think about treating the disease. The realization was treating the virus alone isn't enough.
Because of the cytokine storm.
Right. If the virus has already triggered that massive immune panic and your immune system is actively destroying your blood vessels, just stopping the virus won't turn off the immune system's panic mode.
You have to treat the collateral damage.
Yes. So researchers tried combining ribavirin with high-dose steroids, specifically methylprednisolone. Steroids act as a powerful immunosuppressant. They calm down that hyperinflammatory response.
They turn off the alarm.
And the clinical results were striking. Patients receiving the high-dose steroids saw their dangerously low platelet counts begin to reverse and increase within just 36 hours.
Which is literally the difference between life and death when you're bleeding out.
Absolutely.
But still, relying on a controversial antiviral and heavy steroids isn't a long-term solution. How are researchers planning to stop this in the future? What is the actual cure going to look like?
Well, to understand where we're going, you have to appreciate how bizarre the history of trying to build a vaccine for this has been.
Oh my gosh, bizarre is putting it mildly. I was reading the section on the history of vaccines, and my jaw actually dropped.
It's quite the story.
Between 1974 and 1996, Bulgaria actually used an inactivated vaccine for humans. Do you want to explain how they made it, or should I?
They derived it from the brains of suckling mice.
They were injecting mouse brains into people. That is insane.
It's wild. Scientists would infect newborn mice, allow the virus to replicate heavily within the brain tissue, harvest that tissue, chemically inactivate the virus, and use that as the base for the vaccine.
And it was never approved by the FDA or the European Medicines Agency, right? I have to assume injecting animal brains into a human arm is a spectacularly bad idea.
The safety concerns were massive, to put it lightly. When you introduce foreign nervous system tissue into a human, you carry a severe risk of triggering an autoimmune response.
Like your body attacks itself.
Exactly. The patient's immune system might get confused and start attacking the myelin, the protective coating of their own nervous system.
Yikes. So mouse brains are firmly off the table. But the research points out that making a modern, safe vaccine for this virus is uniquely infuriating because of its genetics.
It is a nightmare for vaccinologists. The virus's genome is divided into 3 segments: large, medium, and small. The problem lies entirely in the medium segment.
Well, the medium one.
That segment encodes the surface glycoproteins, basically the external spikes on the virus that our immune system uses to recognize and attack it. But that medium segment is incredibly unstable. It has a sequence divergence of up to 31%.
Meaning it's constantly mutating and changing its appearance.
Right.
It's like putting on a different disguise depending on which subclass of the virus you catch.
Yeah.
How do you train an immune system to look for a criminal that changes its face every week?
It is an incredible biological hurdle, but there is real momentum right now, thankfully. We've recently seen a DNA-based vaccine that successfully protected macaques, non-human primates, from the disease.
Okay, progress.
And even closer to home, the University of Oxford has developed a viral vector vaccine called ChAdOx2 CCHF. It is currently in phase 1 human trials in the UK.
That's amazing. But vaccines only help you before you get bit, right? What about the people already in the ICU? The clinical review gets into some wildly futuristic treatments using monoclonal antibodies, or mAbs.
Yes, the antibody research is fascinating.
And it breaks them down into neutralizing antibodies and non-neutralizing antibodies.
Understanding the difference between those two is critical to how future treatments will work.
Let me workshop an analogy here and tell you I've got this straight. Neutralizing antibodies. I'm picturing them like bouncers at a nightclub.
Okay, I get it.
They bind directly to the virus's surface and physically block it from entering your healthy cells. The virus simply can't get past the velvet rope.
That is an excellent comparison. They mechanically stop the infection at the door.
But the non-neutralizing antibodies, the research mentioned one specifically called 13G8, they don't act like bouncers. They bind to a completely different part of the virus, a secreted protein. So they don't block the door at all.
Right, they have a different job.
It's more like they are pulling the fire alarm. The virus still gets in, but the antibody alerts the body's natural killer cells to rush in and destroy the infected cell immediately.
Exactly, and the data backing up that fire alarm approach is stunning. Studies in mice demonstrated that prophylactic administration of that specific 13G8 antibody resulted in a 60 to 90% survival rate against a completely lethal dose of the virus.
Wow.
It actively stopped the virus from spreading into the liver and spleen to cause those fatal hemorrhagic lesions.
So we have bouncers and we have fire alarms. Why not just use both?
That is exactly the holy grail of current research.
Yeah.
Scientists are developing bispecific antibodies. The review highlights one in particular called DVD121801.
Catchy name.
Right. But it is literally engineered to bind to 2 different targets on the virus simultaneously. It creates a highly synergistic, inescapable defense.
It's like a bespoke microscopic weapon.
And in the future, it won't be used alone. The protocol will be a layered defense. We're looking at combining these advanced bispecific antibodies with a new generation of highly potent antivirals.
Like what?
The review points to 2 experimental drugs, favipiravir and 2-VET-DFC.
Which are essentially the upgraded modern versions of ribavirin.
They target RNA replication, much like ribavirin, but with vastly greater efficacy and significantly less toxicity to the patient. More importantly, laboratory studies show that favipiravir and 2-VET-DFC act synergistically.
They work better together.
Exactly. When you use them together, they suppress the virus's ability to replicate far more effectively than either drug does on its own. So the ultimate treatment protocol of the future will likely look like this. Block the virus from entering cells with advanced antibodies, utterly shut down its replication with combined antivirals, and manage the immune system's cytokine storm with targeted steroids.
A complete tactical takedown of the pathogen from every possible angle.
That's the dream.
Okay, if you're listening to this, we've covered a staggering amount of ground today. We've tracked this highly adaptable, terrifying virus from infected ticks hitching rides on migratory birds, high above the 50th parallel, down into the bloodstreams of silent, asymptomatic cattle.
It's a huge journey.
It really is. We've explored how it uses tick saliva to sneak past our defenses and trigger a cytokine storm that systematically dismantles our vascular plumbing. And we've seen how human ingenuity is racing to catch up, abandoning 1970s mouse brain vaccines in favor of bispecific antibodies and targeted molecular antivirals.
It is a profound reminder of how deeply interconnected human health is with global ecosystems. I mean, a bird's flight path in North Africa can literally dictate a medical emergency in a Spanish hospital.
It's humbling, honestly. But before we let you go, there is one final lingering mystery buried in the margins of all this research, something we really want you to mull over.
Yes, we know that humans are the only species that suffer this devastating, fatal hemorrhagic fever. Domestic animals, cattle, sheep, goats, they act as silent carriers. They don't get sick at all.
Which is still so strange to me.
It is. In fact, some livestock populations show a 25% seroprevalence. That means a quarter of the herd caught the virus, cleared it from their system, and developed antibodies without ever showing a single symptom.
And the data also shows there are 0 documented cases of a human catching this twice in endemic areas, even though we still don't fully understand the mechanics of how long our protective immunity actually lasts.
Which leaves us with a truly fascinating question. What if the ultimate cure for this disease isn't initially going to be synthesized in a multi-million-dollar laboratory?
Where else would it be?
What if the master key is already locked inside the biology of a completely asymptomatic infected sheep just standing in a field?
Oh, wow. If we can crack the code of exactly why the virus lives peacefully in a cow or a sheep, but completely destroys the plumbing in a human, we might just figure out how to disarm the disease entirely.
Exactly.
Sometimes the most advanced medical breakthrough might just require us to look a little closer at the sheep. Thanks for taking the deep dive with us.